I'm going to introduce Dr. Mike Pearson. He's a senior biologist and principal at Pearson Ecological. Mike is a registered professional biologist with over 30 years of experience in fisheries ecology and management of species at risk and the design, construction, and monitoring of habitat restoration and enhancement projects. Recent work has focused on flood resilience and standards for monitoring, and I'm looking forward to hearing Dr. Pearson's talk today, which I expect will be informed by Mike's extensive field-based knowledge on implementing solutions for salmon in the Lower Fraser Valley and area. So Mike, turn it over to you.
Hey, good morning, and thank you very much for the opportunity to speak with you. I'm going to be focused mostly on what happens after you collect the data. Sort of on standardizing analysis and reporting and sharing of the information, and ways that can help us all develop better processes going forward. Okay, next slide, please. So why we need to standardize these things? And some of this has been covered by previous speakers. So as Mike Bradford pointed out, often the people that are doing the monitoring are going to be different than the people that did the actual restoration work. Even if they're not, over the course of these longer monitoring periods, which we're realizing are necessary to really understand the effectiveness and the impacts, there's unlikely to be anyone on the team at the end of it that was on at the beginning. The second one is accessibility, and this will become clear, I think, as later on in the presentation. But how we manage data, how we talk about it, and how we disseminate it is important in terms of accessibility. That's important for reconciliation. Indigenous groups and communities have a very strong interest in restoration, of course, and often don't have the in-house, expertise. We need to be talking about results in ways that everyone can understand, not just Indigenous communities, but general public, the nonprofit organizations involved in this. And that will give a broader perspective, because when you get all of those viewpoints in rather than just the technical, it's going to make things stronger in the long run, and we will understand it in a broader way. And as was pointed out by both Karen and Mike earlier, meta-analysis is important. Being able to scale the results from individual sites and projects to understand what goes on between regions, what are the differences in project effectiveness between regions, what generalize across specific projects, and look at which types of projects are effective and which less effective. Basically being able to scale up. Next slide, please. So I've been working with the last number of years on these kinds of things. First with Pacific Salmon Foundation, making a lot of this material, and then more recently been applying it, sort of testing it with the Resilient Waters Project in the Lower Fraser Valley, which is looking at flood infrastructure. And so I'm going to talk about three things, sort of the indicators, which I think everyone's pretty familiar with, and sort of what makes a good indicator and how we can use them and generalize with them. Calculators, which are something we use that allows non-specialists to do analyses. You don't need graduate work in statistics to be able to analyze the data when it's automated. It also ensures that it's done the same way every time. But making those so that there's some flexibility in them, as has been emphasized in other talks, that's really key to allow these approaches to be applied with different target species and with different project types. And then reporting. I think it was Karen referred to sort of the dusty consultant's report sitting on a shelf. A typical consultant report for these things can easily run 100, 200 pages long. Not many people read those, and certainly not any non-specialists. So a report card format, and that can be instead of or in addition to those kinds of reports, are much more accessible. So they need to be brief but dense. So they've got a lot of information packed in them. Lots of very clear what the results are, and and for the indicators, it's important to use clear thresholds. And I'm not talking about if it's above a certain level is good and it's below a certain level is bad. That I'll explain a little more later on, but more sort of reference points or benchmarks that you can compare to begin to talk about things and compare them. Plain language is important, right? We in the business talk about CPUEs and LWDs, and that is largely unnecessary for communication. CPUE is catch per unit effort. We describe it in the report cards as average number of fish per trap. It's the same thing, but one's understandable by a broad audience, one is not. And again, standardizing the presentation of the report cards. Okay, next slide. So here's an example of the indicator thresholds. And so the water quality ones I've done because they're fairly broad-based. They're used. There's national and provincial standards for these, and lots of literature on them. But to use maximum temperature minimums, oxygen levels for eggs, and rate them into these different threshold categories or bins, right? So green, blue, yellow, and red. Red being generally the worst for fish. These particular values are set up for salmon in the Fraser Valley, particularly coho, which is and Chinook, which is what we're focused on. But they could be adapted for different target species as need be. Less common used are ones, the fish community thresholds we use. And again, these would be regionally specific, but the number of native species we catch at a site, the percent of indigenous species, what are the percentage of salmonids, are there species at risk present? One that might not be too intuitive at first is the percent catch of three-spine sticklebacks, which are actually a great indication of predation pressure. If you're out there using G traps, you're catching all your little salmon, but you're not catching the predators. The larger cutthroat trout, the pike minnow, and that kind of stuff. So, the proportion of your catch at three-spine stickleback can give you an idea of that. If you're catching hundreds per trap, likely predation pressure is pretty low. If there's one or two, because these sticklebacks are very tolerant of environmental conditions, if there's very few of them, often it is, or most often, it's predation pressure that's driving that, which gives you some insight into what the juvenile salmon are facing as well. The number of introduced species, the proportion of traps that are empty. So you see that these indicators, most of them are generalizable across different project types and with some moderate tweaking among regions. Okay, next slide. Here's an example of the water quality indicators applied to Tilbury Slough down in Delta. This sampling was done in April. So as you would expect, the temperature was pretty good across the board. You can't really see the reach names on the map, but the top line here is just an aggregate of all the data across all the reaches. Reaches 1 and 4 are outside the dike. So at this end and up at this end. So habitat that's directly connected to the Fraser River. 2 and 5 and 3 are confined between dikes. And you can see a considerable difference there. There's, you know, outside the dikes, oxygen is generally quite good. Inside, it varies, but there are severe hypoxia in you know, in portions of the reaches, or in some cases across the entire reach. Okay, next slide. This is the same outing, the same sampling session in Tilbury, but looking at the indicators for the fish thing. And this, you see the same kind of thing in reach 1 here, and in reach 4 at the far end, which are outside the dike. You've got more indigenous species, a much higher percentage of catch of indigenous species. Some monitored proportion of the catch is low across the board, but it's actually 0 inside the dikes. So you can start to tease things out, and it's in a very understandable manner. And again, I'll come back to these color codings. Red is not necessarily bad. I haven't shown it, but we also work in the Upper Pitt River, with the best coho habitat I've seen in my life. But there are 4 species in that habitat. So being red or yellow doesn't necessarily mean things are awful, just that they require some explanation. And this, I think, is important when we're talking about using thresholds, particularly in a regulatory context. There's going to be a lot of suspicion and fear among some proponents that, you know, we need to be above or below this. And I think stepping away from that and not treating them as, you know, as pass-fail kinds of things is an important consideration. There's things that require comparison and some explanation as to what's going on. Sometimes they will be of concern, but sometimes they won't. Okay, next slide. Now the calculators. So what I'm going to go through in some detail is the fish indicator calculator. The one that we've developed is just an Excel, which I think has some strengths in terms of it's, you know, on everyone's computer, so it's very accessible. Lots of people can manipulate or customize it to some extent for different regions as necessary. But the same approach could be used in proprietary apps or what have you. So, I mean, the first page of it is just simple introduction to the site, when it was sampled, who was there, what were the reaches. As we're going through these pages, the green cells are spots for data entry, where users can enter things. Orange cells are calculated. So next slide. So this is the data. You don't need to see it all. Know that each line of data here is a single trap. And, you know, it has the basic data, the watershed, UTM coordinates, water quality information that we catch. We take temperature and oxygen, conductivity at every trap, and then just the number of each species that we caught in each trap. Now we collect the data using a different app called FilePro, which is we use iPads, and it's just a very user-friendly front end to it, very fast and easy to collect data in the field. And then we export that into an Excel spreadsheet with exactly this order of fields or columns in it. So once we clean up the data, it's a very simple matter to just copy that exported spreadsheet into the green cells in this calculator. Just a couple clicks and it's in there. And then the calculator is set up with kind of formulas all linked all through it, so that the minute the data is in there, all of the indicators are calculated, and a lot more besides the indicators. And all the tables are generated that we're going to use in our report cards. So very automated and easy to use. Okay, next slide. Well, this is a calculated sheet, and it just figures out from that spreadsheet that we copied in exactly how many traps of different types were set, how many were empty, how many seine holes we did. That's the top panel. This panel is just, and it's all by reach again, all the different reaches at Tilbury Slough here. The next panel down is what the catch was. How many coho salmon did we catch in each one? Chinook. And this lists every fish species, native and introduced, that is known in the Fraser Valley. Next slide. So then for the calculator table, this is an exact copy of that last one. And all that we do is delete all the rows from this one that are blank. And you end up with a little table that by reach exactly what you caught in each one. Very easy to use. And again, really minimizes errors and the sampling effort in the table below. So the purple is the sort of the third color we use. The purple just indicates that it's data that gets copied into the report cards. Okay, next slide. Okay, this next worksheet is entirely calculated, and it groups things. So the first one is the number of species, again by reach. So the number of indigenous fish, the number of salmonid species, the number of introduced fish species, the number of species that are predatory on salmonid juveniles, the number of species at risk. We also include amphibians. And then there's the number of individuals caught. Again, the total number of indigenous fish, the total number of salmonids, all of that. And then there's also catch per unit effort of all those groups. So we don't use all of this data in the report cards, but we calculate everything. And then the idea is that we can cherry pick what we want out of this to use for the indicators. And this is important because for different projects, we might be say, building or working on a habitat that's for sailor sucker, a species at risk, or one for Chinook salmon. And those would have those species-specific indicators, or catch per unit effort, or average number of fish per trap, as we put it, to be put into the indicator table, which is on the next slide, please. So here's the indicator table in the calculator. It's not color-coded. I'm sure that could be programmed. That's beyond my skills at the moment. So we color code them by hand when they go into the report cards in relation to those threshold values. So here where there was 8 fish, our threshold for green is 6. So it's above that, that would be green, et cetera. And all of these would be color coded accordingly. But again, this data can be just taken directly from the calculator and put into the report cards. And again, these list of indicators, I think there's, some of them that would be pretty standard across, most projects, certainly in British Columbia, where we're working, most of the projects that we're dealing with involve salmonids. So the proportion of the catch that's salmonids would probably be a broad indicator. Other things like that would be more customizable. But again, if we've got all of it in the calculator, then the same data can be used in future in different ways for different purposes. Next slide. These are just a couple of other tables that the report card does. It or the calculator pulls out all of the species and then has, the indigenous halcomelum name, scientific name, the provincial code for the species, and its status, whether it's introduced or indigenous, whether it's a species at risk and what its specific status is with Kasiwa, Coursera, the province. So again, a little table that would go into the report card. Water quality, we measure oxygen, temperature, and connectivity at every trap location. So this just summarizes that in terms of minimums and maximums, makes a little table out of that. Okay, next slide, please. Here's an example of the report card. So the top little table is, again, the who, when, where kind of information. That it was at Tilbury Slough, who was on the sampling crew, the month and the year. Here's our indicator table, all color-coded. The list of species encountered. So again, brief, dense, and written without jargon. A little map showing where all the traps were set. You'll notice the blue dots are where we caught salmonids. So they're outside the dikes there. These two little green, I haven't talked about yet, but those are locations of traps where we put oxygen loggers inside the traps. And so we get an oxygen log for just a couple of days hourly measurements. And those graphs would go into the report card as well. Little box of just point form interpretation. What does this data mean in very plain language? There would also be another couple of pages I haven't shown here that would have the actual catch table, sort of by reach, what the different species, how many we caught, photographs of a representative photograph of every species as well. Okay, next slide, please. And so I've concentrated most on the fish one, but we've also developed these for riparian areas. So we do sort of census plots. They're like 250 square meter plots in restoration sites that we were monitoring. And indicators are like, we focus on trees, shrubs, and ferns. So things that are that there's a manageable number of species for non-specialists to learn and to identify. And it's the trees and the shrubs that are functionally the most important to the aquatic habitat. So we'll look at the number of, or calculate the number of native, total number of trees, shrub, and fern species, their density per 100 square meters of shrubs and of trees, the density of trees over a certain size. We've picked 10 centimeter diameter at breast height, the density of snags and large woody debris over a certain size, the maximum tree size, the proportion of canopy cover, which we use photographs and an automated software to sort of calculate those percentages, the number of introduced species that are present and their prevalence, whether they're absent, present, common, or dominant. And again, those have indicators associated with them and, thresholds associated with them to sort of rate and compare the sites. Next slide. Same with water quality. This is one for temperature logger data over a couple of years. And so it calculates maximum temperatures, minimum temperatures, but also durations. These are temperature duration curves where it's just you take all the data instead of presenting it chronologically as the logger, the first graph shows. It's just presented. It's sorted in order of the temperature itself. So the slope of these graphs gives you important information and how much time it spends in different the water is in different temperature regions, I guess. And so we relate that then to growth conditions, in this case for salmonid. So this table below here shows that, it's dangerously warm is above 20 degrees Celsius. How many days cumulatively did it spend above that? So often if it says 5 days, it's not that it was 5 days in a row above that. It was probably an hour or 2 hours above that all through July or August each day. But it gives you an intuitive way to compare sort of the temperature profiles of these things. Where if you just look at the logger graph itself, it's pretty limited information. Really, you're going to focus on the peaks. And, did it get too warm? It would be one of the big things, or did it freeze? But being able to break this down in understandable ways is important. And again, communicating it very briefly. And another thing I'd point out with these report cards is the ones I presented are for individual sampling sessions. But you can see that you could build these to compare different sites. You could add control reaches onto them, which we do in some cases, or you could also build them across years and look at how the same indicators change from year to year at a different site or seasonally. So they're flexible in that way, scalable, I would say. Next slide, please. Another example of habitat report cards. These are ones, this one is for a specific site that in the Resilient Waters project. And it's not really based on a calculator, but more on sort of survey data or just a reconnaissance survey, really. Sort of a summary of what the site conditions were. Did we see any restoration opportunities? The indicators would be like the current connection to the Fraser. Is it connected, or is it behind a dike? Is there a floodgate? What kind of floodgate is it? How long is the channel? Fish accessible inside the gate? What potential species would use it? That kind of thing. What's the condition of the riparian? What's the habitat complexity in qualitative terms? Those kind of things. So again, a communication tool and ways to quickly summarize what's going on at a site. Okay, and next slide. This is just the second page of those, which break it down by reach with photographs of each one. You can see that our floodgate structure here is in pretty rough shape in the first picture. But, and I believe the last slide, please. So thanks very much. I appreciate the opportunity to talk, and I'm looking forward to the panel discussion.